Welcome to Whose Gene Is It Anyway, where the species are made up and the ethics are made up and everything doesn't matter. Uh, so, welcome to another episode. I think this is going to be hopefully a fun one. Um, as usual, the way this show goes is I'm going to pick a ridiculous idea out of a hat. This is the hat. And many of these ideas have been submitted by you. We're going to pick an idea, and then we're going to spend the next hour or two designing a piece of DNA that should accomplish the task on the little piece of paper. So, I think it's about time we get started. So, before we do anything, I'm gonna have to draw an idea. I mixed these right before the show. So, let's go ahead, give it a little mixy mix. Ooh. It ran away. Okay, let's see what we got. Oh, this is fun. There were a couple of requests for this one. Capsacin yeast. We're gonna make some spicy boys today. This is gonna be fun. Okay, so before I forget, like I inevitably do every single frickin' time, I'm gonna go ahead and change the name of the stream. Just so people know what we're doing. Now I gotta remember how the hell you spell capsaicin. There we go. Capsaicin. So how's everybody doing? How's the lock-in? Hope you're all staying healthy and happy and not going too crazy. And the nice thing is, once we've got capsaicin yeast, I mean, you could put this in anything. Spicy beer sounds, personally, I think it sounds awful. I hate spicy, but I know people are all about the spice life. Anyway, okie dokie. Stream name has been changed. Okay, so before we get into it, I want to do a little update on how things are going on my end. So, as I said on Monday's stream, I've been working on the spider silk project for the last week or so, amongst other things. But I mean, that's been the prime focus. And so when we last left off, I had managed to isolate the silk gene from the piece of DNA that we'd ordered, and was working on building it into a plasmid that works in 𝐸𝐵𝐢. The reason being that I've been having a ton of problems with getting the DNA into the yeast, and I don't know why. I did 8 transformations on last Sunday, and not a single one of them worked. I just checked them today, and still nothing grew. So if nothing's grown at this point, nothing's going to grow, and that really blows. And I honestly do not understand why it isn't working. I've done yeast transformations before, and it's worked fine, so I don't know what changed. That's pretty fucking frustrating. So in the meantime, I was trying to build the 𝐸 𝐵𝐢 version of the spider silk project, and that is also not working. In this case, for a different fun reason, which I'm yet to figure out. I think there was an issue with the donor plasmid that I used. So we talked about this last time, but I'm taking the spider silk gene out of the plasmid that the manufacturer made for me and putting it into a different plasmid. Something was wrong with that plasmid I'm putting it into. I may have grabbed a tube that was, I don't know, didn't have the DNA in it or something, and I don't know why. But yeah, so that's what I've been struggling with. Oh boy, is it very frustrating. So I've got to kind of put that project down for a couple of days. I'm going to give it another go, probably next week. But yeah, as it stands, very frustrating. I think I know how to fix it. but I'm basically back to square one if I'm wrong. So, not fun. Not super fun. But we're going to have a very fun day of making some capsaicin yeast today. So I think that's going to be a lot of fun. So, kind of make up for it. Okay, so let's get to the spicy boys. Ooh, actually, there's one last thing I just got to do really quick, which it occurs to me that actually, I don't know if I can do that. I don't know if I can do that live. Fuck it, I'll do it next time. It's not a big deal. All right, let's get into some designing, shall we? Okay, so we are, as usual, back to our designing phase. So this is the program that I use for all of my genetic designing stuff. Oh yeah, see the goal still is up. I don't know if I can fix that while it's live. Well, anyways, as you can see, we hit our goal from the fundraising goal for the last time. So I was actually able to buy my fluorometer, which is super exciting. That should arrive in a week or two. And for those who don't know what a fluorometer is, it's basically a special device that lets you figure out exactly how much DNA or RNA or protein is in a sample. And this is really, really important so that, like, a lot of protocols require that you add a very specific amount of DNA. It's like, I mean, imagine trying to bake and you have to eyeball the flour, but the flour is invisible. Like, obviously that would be very difficult. So, you know, it's important to know, you know, what's up. So yeah, that's exciting. I was going to change the goal today to be a new goal. I want to do a little bit of a fundraiser for a capture card, because as much as I've been enjoying these live streams, I would really love to be able to live stream some actual like work, like so actually like tinkering in the lab or cooking or, you know, whatever the case may be. So I was going to do that, but I had an issue with my mic right before getting started where just no sound was going through. So that kind of took priority, and so I didn't get that adjusted, but that's fine. All is good. All is good. The difference between a UV vis nanodrop and a fluorometer is a fluorometer you have to add a dye to your sample to make it work, but it actually is more sensitive because of this. So like a nanodrop, like you could put your sample on it and it might tell you that there's DNA, but it could just be contamination. Like there's no way to know. I mean, there's a way to guess, but there's no like concrete way to know, like, is the thing that it's picking up actually DNA or is it just some like other garbage that was in there? Am I going to be doing a bread video? Yes, that's the next video that's coming out probably. So yeah, this was the plasmid we made last time that makes cellulase in yeast. So it's like yeast that you could grow on like toilet paper, which sounded fun. As I said I would last time, I have updated the GitHub. So all of I also updated the milk and egg plasmids, so they're now ready to be submitted for a quote. I'm holding off on actually submitting that so that y'all can have a look at the DNA for those of you who are competent in the ways of messing with DNA and see if I missed anything. So I'm leaving this up for a couple of days, and then I'm going to put the quote in, and then those are going to get ordered. The other two are now in there as well, so that's exciting. So let's look up capsaicin. Capsaicin. Did I spell capsaicin wrong in the thing? I did. I knew it was a weird one. Capsaicin. That's annoying. Okay, well, fix that. Okay, I gotta do all this shit again. Okie dokie. Capsacin biosynthesis. Rightio. Let's see. Where does this come from? Oh, really? It comes from... Interesting. Very, very interesting. So the things that this is telling me is it's a caffeic acid derivative. And to my knowledge, yeast doesn't make caffeic acid on its own, so we'd actually have to include that. But this does remind me, and I had capsaicin yeast written down, but I'm almost tempted to do capsaicin tomatoes. Yeah, because I mean we've done yeast for every single stream so far, and I mean I'm not opposed. Like we're gonna have a little look see and see how difficult this actually would be to do in yeast. But there was a really great paper, since tomatoes and peppers are in the same family. Technically, tomatoes have the genes for capsaicin. They're just turned off, or at a level that's so low that they just don't function. So I'm almost tempted to maybe switch it up a little bit and maybe do tomatoes. So I'm just having a little look around. Oh, really? It comes from vanillin. That's really interesting. Okay, so I'm looking at this. So it goes from caffeic acid to vanillin, which is like vanilla. So that's really, really interesting. Like really, really interesting. You basically would have to do vanilla synthesis to be able to make capsaicin. So it looks like capsaicin is actually pretty intense. So I'm not sure. I kind of want to. Yeah, this was the paper I was referring to. So you can see that these are the different genes that. Well, actually, this is really quite helpful. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9 genes. Wow, that is that's a chunky boy. Yikes. So it seems like the only difference between a sweet pepper and hot pepper is this CS gene, which we'll find out what that actually does at some point. So it looks like tomatoes are only missing. So it says promoter replacement, which to me says these are there, they just aren't functioning for some reason. So you could probably, like, if you can go in, turn those on, that'd be good. And then it should do it, because it seems like as long as you've got this CS and whatever this BCAT does, it should just do it. So with a sweet pepper, it's a difference of 1 gene, whereas with a tomato, it's a difference of 2 genes, and also just the levels are too low. So like it wouldn't be very spicy, but spicy nonetheless. Ooh, thanks, Ron. I may take you up on that if we decide to go. So what do you guys think? Should we do spicy yeast, or should we try and do spicy tomatoes? I'll leave this up to you guys, because I'm fine to go either way. Because I'm seeing about oh wow, okay. It seems to be basically like I think we're leaning towards tomato. I'm seeing some yeast, but most people seem to be saying tomato. So I think we're going with spicy tomatoes. So I'm gonna actually I'm gonna change the stream name then. Ooh, somebody kicked in or did something. I didn't see that happen. Oh, thank you. Well, thank you, Anthem. So yeah, I'm just gonna go ahead and change this to tomatoes, because I'm kind of yeasted out. Been doing nothing but yeast for the duration of this stream. It's gonna be nice to do something different. Okay. Is this yelling at me about the spelling? No. It's trying to autocorrect capsaicin to captain, because autocorrect does not know the science. Okie dokie. Let's go back over to here. Cool. Can you make carnivorous tomatoes? tomatoes are already carnivorous. I don't know if you guys knew this, but tomatoes are carnivorous plants. This is a little known thing. All the little hairs on the stem of the tomato, if a bug touches them, it releases stuff that kills the bug, and then the bug drops down and dies, and then fertilizes the soil. So yeah, tomatoes are technically carnivorous. Okie dokie. So since we're gonna, we got it. So let's. I mean, we should just open up this paper if we can. Let's see. How did they do this? I think this was just like an analysis. I don't know if they actually did it. I think maybe the easiest way to do this, rather than doing, I mean, we're gonna have a look at this. Oh man, oh, this is exciting. Is it actually a locked paper? I think it is. I think it is. Oh man, this is a first on this stream. We get to demonstrate my favorite website. So this paper is behind a paywall because they're bastards. So we get to do one of these, 𝐎𝐵.tw/ and then, okay, so what I'm doing is, this is Sci-Hub. It is the greatest site ever. So what you do is you just go sci-hub.tw/ and then you just like post the DOI number. This is the DOI number here. Ignore the DOI.org garbage. You just need the number at the end, and you just give it a little pasty paste, and you click go. And then, ta-da, you've got your paper. Yay! Isn't this fun? Yeah, Sci-Hub is the greatest thing to happen to science in 100 years. So that's pretty damn cool. So this is fun. Yeah, capsaicin is a really interesting molecule. It's I never knew that it was a vanilla derivative. See, this is what I've really been enjoying about these streams. Like, it's forcing me to do stuff that I would just never look into. So it's been pretty cool. It's pretty cool. Okay, so here's the thing. So we're missing. Oh, now it actually tells you what the genes are. How wonderful. Okay, I mean, this makes sense. CS is capsaicinoid synthase. So yeah, it's the make capsaicin gene. And then BCAT is branched chain amino acid aminotransferase. Not sure what that does, but hopefully, okay, that's not actually in this paper. Okay, well, at least we've got our list of genes, so we can look that up separately. Ooh, somebody kicked in. Well, thanks, Stookie. Okie dokie. What do we got? Towards hot tomatoes. Okay, this seems to be the thing that we actually need. So I think in my head, the easiest way to do this, considering that even though the levels are low, they are present, probably the easiest way to do this would be to just provide a fresh copy of both of these genes, the capsaicinoid synthase and branched chain amino acid aminotransferase. That's going to get really fun to say. We're going to call it BCAT. So yeah, if we just provided fresh copies of these genes to the tomatoes, then it would immediately be a little spicy. It wouldn't be very spicy, but that's the sort of thing that you could either find a variety that has higher levels of all of these things, or you could do some like mutagenesis-ish to do that. But I mean, the big one is just get the new gene in there. So this is exciting. This is really, really exciting. So there's a lot of different ways to modify plants, but agrobacterium is essentially the go-to, which for those of you who don't know what agrobacterium is, it is, if you've ever seen like a tree with like a big weird looking tumor thing on it, That's what Agrobacterium is. Agrobacterium is this really interesting, bacteria. Yeah, so this is an Agrobacterium infection. So basically, what Agrobacterium is this weird bacteria that infects plants. But it doesn't just infect them; it actually genetically modifies the plants to basically do something that it wants. So usually, I believe it induces some amount of like vascularization to the area, so it causes this like weird growth where it's sort of like diverting nutrients to that part of the tree for the bacteria to grow. And this can like anytime you see a tree that's this weird like mottled garbage, that's what it is. And it also can upregulate various plant hormones, because I mean plants are really just, I mean, to steal a line from a friend of mine, plants are just hormone distributions over space and time. So if you mess with a hormone somewhere on a plant, you get all kinds of weird things happening. So in this case, if it starts like boosting like gibberellic acid or something, you can end up with like this huge weird growth. So what scientists have done is they've taken this bacteria and they're like, okay, cool, this thing can get DNA into plants really easily. That's literally, it's like, that's its thing. So what you do is you remove all of the pathogenic garbage from its DNA, and then you design a piece of DNA that will be put into plants, but basically like won't harm them. And so what you do is you callus. So what you do is you grow one of these. I call it a plant booger, but it's called a callus. And so basically what this is you take a little piece of leaf usually, and you put it on a special petri dish that has hormones in it that basically make that little piece of leaf devolve into stem cells. And once it's in this like stem cell state, you expose it to the bacteria and let it like cook for a couple of days. And then after like 3 days, you basically treat it with a really high concentration of like an antibiotic to kill off all the bacteria. And so you end up with a modified plant. So what you could do, and you can actually do this with tomatoes. I have a friend who's done this, and he actually made a tiny tomato just through messing with it. So yeah, that's basically how this would go. So what we're going to end up having to do is design a bit of DNA that would work with Agrobacterium. So we need the 𝐴𝐫𝐢𝐠𝐢𝐧𝐚𝐥. I believe this is the plasmid that we want. Yeah, Agrobacterium binary vector. That's exactly what we want. Kanamycin resistance. Okay, what do we got here? There's your multiple cloning site. Unfortunately, this is all new to me because I never use this. Okay, replication protein, stability protein. But where do you actually put it? Like, where do you put the DNA? Hey, plant people, how does this plasmid work? Where's I see a lac promoter, but a lac promoter is not. Oh, wait, what is this? Oh, I assume it goes here? No, wait, that doesn't make any sense. If it went here, that would mess with the antibiotic resistance. So where do you put it? Let's see what 𝐴𝐫𝐢𝐠𝐢𝐳 is. So yeah, these plasmids are built to work with Agrobacterium. So the first thing that you would do is you basically take this piece of DNA and you transform it into you transform it into the Agrobacterium, and then you just expose the plant to the Agrobacterium, which in theory should be fairly straightforward. don't know what I'm looking at. Interesting. Oh, 𝐵 has no promoter. You need to add a promoter for it. You add it from another plasmid to add the 35S promoter. Oh, okay, yeah. I mess with that. That sounds good. So let's go back then. Let's use 𝐵 2300. Because why not? No, I just want the map, please. Well, so do you have to add a terminator too? Do you just like pop it open? Like, what's the... Okay. So is the idea that I would just like cut it open here and then just stick in the whole promoter and gene and terminator? Am I understanding that right? Is that how this goes? Because I mean that seems the easiest. I mean, I'm seeing an 𝐎𝐫𝐢𝐠𝐢𝐳 site, so you just pop the fucker open. Usually transform to then 𝐵. What's PSAT 6? PSAT 6. Okay. But I gotta take this call really quick, so I'm gonna just pause the stream. I will be right back. Okay, I am back. Sorry about that. Been waiting on that call all day. Okay, so we were looking up the PSAT 6 thing. What is this? What is this? What's it do? Oh, okay. Oh, YFP fusion. Okay, this makes sense. I see what you mean now. So the idea is you would get this. I mean, this is sort of a different strategy than the way that I do things. So I think what Ron was suggesting is you would get, if you were doing this the old school way, you would buy this plasmid, and then you would cut it open right here, where all these restriction sites are. You'd stick your gene in, and then by the looks of it, oh wow, that's actually a really convenient cut site since I have both of those enzymes. Yeah, so then by the looks of it, what you would do once you've got your gene in here is you would cut with AGEI and either 𝐎𝐻𝖏𝑚 or 𝐎𝐫𝐢, and then you would like cut this whole chunk out and then stick it into this guy, presumably right here. I don't know if these have compatible ends, but I mean, you could add them. That's not really a big deal. But yeah, the way that I don't mess with that if I can avoid it. Honestly, if I never need to touch a pipette to make a piece of DNA, I do. The fact that I'm having to do it the hard way with the silk is basically like a test of my patience, because I'm only going to give it another like week or two, and if it keeps failing, I'm just going to say fuck it and buy it. Because it is so much easier to just pay a company to make this for you. Yeah, PCR out the DNA. Yeah, sure, fine. If I never need to do PCR, I avoid it if I can. I hate PCR. I ran like 4 of them earlier, like in one day earlier this week, just because it kept failing for no obvious reason. So, yeah. We're gonna go with this. This is our backbone, because download. Cool. So now let's go over to here. Open up a new. Let's make a new folder. Spicy tomato. Jesus, tomato. I can spell, I swear. Yeah, so the paper you're referring to is this guy. They technically have the genes, but it's easier to just add a new copy than it is to try and mess with the copy that's already there, in my opinion. Yeah, I'm going to take the sequence from SAT6. That's exactly what I'm going to do. So I need to download it if I can. So I gotta find somewhere that carries the actual map. Do they have it on? Where would they have? Because I mean, this place I think carries the actual. I'm gonna just go back to this for a second. They seem to like they carry a picture of it, but do they carry the map? Because there's an image of it. DH10B donor stock. Oh yeah, this is if you want to buy it, I think. The ideas in the hat come from you. If you go to Twitter and submit them via at them or at me and use the hashtag #Who'sGene and then just submit your ideas there. Okie dokie. Well, congratulations, you're the first shitpost of the day, and you're banned. Bye-bye. Yeah, y'all couldn't see that, but there was some quality shitposting in the chat, so you know, I have zero patience for that shit. So Uniprot seems to carry this, but is it going to be in some really derp-tastic format? Is the $1 million question. So let's import that 𝐃𝐚𝐿𝐈𝐴. 𝐃𝐚𝐿𝐈𝐴. Cool, that's done. Oh, did I forget to? Oh no, good, it did. Sometimes I forget to change it so that it gets labeled something ridiculous. Oh, it already is open. Okay, well, I'm going to close some of these other things that we had open from the last time, because we don't really need them right now. Alright, cloning vector. Okay, cool. Is there a way to just... No, there's not a way to just download this. Wonderful. Man, spicy weed would be just the worst prank. What do you hate about PCR? All of it. It's slow, and it fails for no obvious reason, and it's just the worst. I hate PCR. If I never need to do wet lab work, I avoid it. I hate it. Oh, here we go. Sequence. Oh, for fuck's sake. Okay, literally whoever did this needs to, like, die in a fire. That is just the worst. And let me guess, if I click map, it's a fucking picture. Okay, yeah, you know what? I'm sorry, but fuck this. I'm just gonna look this up the old-fashioned way. This is the thing that drives me crazy about bio. Like, if you ever see, if you're ever reading a paper, and they include, like, the sequence of the thing that you want, but they've included it as an image, they need to just get beat over the head with a pipette. It's just the worst. Okay, 35S. Let's see what comes up. Gotta, like, scroll up. Just like... We got F's in chat. Thank you. Cauliflower mosaic virus 35S promoter. Am I reading that right? Yes. No Terminator, though. Okay, well, I'm going to come back to that one, ideally. What if I just look up PSAT6? Do they carry it, PSAT6? I mean, it seems like a fairly... Oh, no. Hard no. Pipette, the weapon of choice. For anyone who's seen Shoot 'Em Up, it's one of my favorite fight scenes in movies. He kills like 4 people with a carrot. Like, long before John Wick did it with a pencil, like, this guy did it, like, killed multiple people in multiple different ways with a carrot. And it was awesome. Okay, I'm gonna grab this one, and then I'll find the Terminator after. Exciting. Okay, let's load this in. This is exciting. We haven't done a plant thing yet. This is nice. It's nice to work in something that isn't yeast. I'm looking for a plasmid that has the cauliflower mosaic virus 35S promoter and Terminator in it. I already found one that does, just so I can copy it out. Nothing. I don't need to actually use the plasmid. I literally just need, like, copy paste fodder. Which this should be the thing I want. Yay, it is. Copied. So yeah, I just need the Terminator now. So if anyone can happen to find one that has the Terminator in it, that would be great, so that I can actually get this done. So now I just gotta find the multiple cloning site. I believe it's right here. What with the fact that it says 22. So you're gonna make me scroll, aren't you? Okay, well, let's just click this then. Lack operator. I need a little further forward. What the hell? Wait, this has the 35S promoter in it. I could have just copied it. Well, that's silly. It's just backwards. So the nice thing with DNA is it can run in either direction, right? So, like, this is why you'll notice that some of these arrows point one way and some of them point the other way. Depending on which, like, side, like if it's on the bottom side or the top side, will dictate which direction the thing goes. So you can actually like really like tight code will actually have this like overlap. So if you read it one way, it's one code. If you read it the other way, it's another code. Like some viruses do that really hard. How hard would it actually be to grow this plant? Growing it would be fine. It's modifying it that's the hard part. Okay, well we're just gonna go with this. Where in the hell? Interesting. 𝐎𝐫𝐢𝐠𝐢𝐳. Okay, I see it. Here it is. Yeah, this really is just in the middle of the... Is this promoter? What is this? Why isn't it telling me? Let's go back over to here. Product. It's a fragment. Oh, I get it now. They're using... For fuck's sake. They're using lactase as a reporter. Okay. So... it's like, why would they... The hell? Why would they do that? That's so silly. So the way that you choose that where the gene is expressed is through choice of promoter. I'm just for right now, I'm just making it run everywhere. So like if you were to eat the stem, it would be spicy too. But I mean, it'd be a little spicy. You got to figure that the like most of these genes don't run throughout the plant. Like if I had to guess, and I could be totally wrong here, but if I had to guess, most of these only run properly inside fruit. So even if this is running or this is running in like the stem, it wouldn't do anything because it's missing all of the precursors. I don't know if it's different numbers of the genes. It could be, but it's more likely just the promoter in front. But also plants are weird, so it could be that they just have 100 extra copies of the gene. I honestly don't know. Yeah, 35S works all the time everywhere. I'm aware. I wasn't really going to go for fruit specific. I just wanted to, make it happen. So this is looking like it's just going to... There's just going to be a chunk of garbage in here. So we might have to just work around that. But that's fine. Yeah, all the restriction sites are in the middle of the multiple... There we go. It's labeled the multiple cloning site, so this is where it's going. Okie dokie. Where do we want to cut this? I'm kind of thinking that we cut it somewhere in the middle. Why is 𝐵AM-HI highlighted? Is my question. So let's just do it. Let's do a little checky poo and see. So these are all restriction sites, right? So these are all places where you can cut the DNA so you can stick new stuff in. But it's always important to make sure that, if you're going to do that, you don't accidentally cut somewhere where you're not expecting. Oh, good. It is a single cut. Oh, wonderful. So yeah, the thing I just did was I just went to this menu, which is digests. And you tell it which of these enzymes you want to check. In this case, I did 𝐵AM-HI. And then it basically checked the code to make sure that there was no other places where this enzyme cuts, and it doesn't. So that's good. We could use this one. I believe I have 𝐵AM-HI. Again, not that I plan on ever making this thing, but I could. Or at the very least, I definitely don't plan on making this by hand. I think this is going to be one of those, like, I'm going to upload it, and if somebody else wants to play with it, they're more than welcome. But, yeah, it's not my jam. Okie dokie. So, we're going to... So, it goes from 𝐶TAG. 𝐶TAG. Wonderful. So, first things first, we're going to delete that and paste in our 35S promoter. Yay! Exciting. I don't know why this still says Laxi. It's definitely not. So that's very cool. Now we've got our promoter in there. Now we need our gene, and we'll deal with the terminator after. If actually, if I'm gonna try and keep an eye on the chat. If anybody can find the 35S terminator, please post a link. So now we need the actual capsacinoid synthase. What do we think? Are we going to get lucky? No, we're not. I probably spelled that wrong. So let's just double check 𝐎𝐵𝐺ene again, because this is one of those. If you can find it on 𝐎𝐵𝐺ene, it makes my life easier. PT35S. Oh, that's true. I do have it after the kanamycin resistance. That's a good point. What's poly-A signal? Is that the same? I'm not a plant guy, so this is like way outside of my usual realm of expertise. Shit, I was looking that up. PT35S. Yay. Yay, Terminator. Exciting. Okay, cool. Where is it, though? I mean, it says that it's there. I just don't see it. I mean, is there another map? No, that's it. 50337. See, when Sassy makes a suggestion, I listen, because I'm consistently helpful. Oh, 337. Poly-A signal. Yeah, I mean, I think that's what it is. I don't know why it's labeled a poly-A signal rather than terminator, but I think that's the same thing. What do you think? Is poly-A the same as terminator? Because I honestly don't know. Let's learn together. Interesting. Single RNA polymerase. What do we got here? Sorry, this is going to be kind of quiet because I've just sort of got to read. It says Terminator. I know it says Terminator, but it also says it's labeled poly-A signal. Well, it says polyadenation signal / terminator, so I suppose it's the same thing. Fuck it, let's go with it. Why not? Can you see my earrings? I mean, they're under my headphones. Wait, which ones am I? Oh yeah, I guess I'm wearing the nuclear ones. I used to have a set that were biohazards, but I mean, I tend to only get new earrings when I lose one earring. So that's kind of fun. Okay, that should be downloaded. So I should just be able to pop that in. We're still looking for the... I kind of got distracted. I wanted this gene. Capsacin synthase. Synthase. This looks like the right thing. Is that a fragment? No, it's complete. Oh, it's tiny. Wonderful. Yeah, 𝐎𝐫𝐢𝐠𝐲1. I'll take it. Is this the thing that I've been using? Yes, it is. Okie dokie. We want T's. We're going to just set this to Arabidopsis. I know that isn't the plant that this would be in, but that's the closest that we got, so we're going with it. Okay, so for those who haven't tuned into streams earlier, the thing that I just did, just to kind of back this up, is first we looked up the name of the gene, so these are all the genes that you need to make capsaicin. In this case, the idea is this would go into tomatoes. We've noticed that there's 2 genes that we definitely need to turn on. The rest are there. They're just at low levels, so it wouldn't be a very spicy tomato, but I mean, you could do that through mutation or breeding. So we were like, okay, cool. We need this gene. So we looked that up. We found it on a site called 𝐎𝐵, which is this. This is 𝐎𝐵. The code that we found here is the amino acid sequence that would actually this is the protein. This is what we want the thing to make. So what we did was we grabbed the FASTA sequence, which is basically just this, but without the numbers and weird spaces. So this is just copyable. And then we need this to be DNA, so I can paste it into my DNA thing that I'm working on here. So I went over to this site, which is 𝐎𝐵𝐢𝐠.ru, and I pasted it in. I said I want T's, not U's, because this is RNA versus DNA. I told it to just give it to me in one line. I hit reverse translate. Little window pops up, and then I just copied the DNA, which is this long line here. So copied that. We now have our 35S promoter. So I can give that a little pasty paste. And while it's still highlighted, I'm going to grab this, so that create annotation. So it's CSY1 capsaicin synthase. Just going to try and spell captain again. It's going to try and spell captain again. Cool, that's done. I'm going to make this maybe a different color. This is the thing. This is the one thing I dislike about benchling. My color choices are just awful. Cool, we can close this. Okay, this needs to... This annotation is too long. We're going to say that this only go... Where... What is this? Where does this actually start? Because I'm sick of looking at it. Base 1008. Cool. So let's grab this. Change this to... I make this shorter. Where does this actually end now? Oh, okay, it really does. Okay, so we're just going to call it 960. 9650. Just because I'm sick of looking at that. There we go. Fixed it. That was driving me nuts. Cool. So we've now got our capsaicin synthase, our CAM 35S promoter. That's a good point. So Ron just said I should check if biosynthesis happens in an organelle, since I would need an organelle localization tag. So shit, I'd forgotten about that. Man, plants are weird. I'm so used to working in bacteria where there's no, or not no, but there's like generally if something's made, it's just made. Like you don't need to do it in a special bucket. But I think, I mean, there's a little graphic on this paper. These are like right near the top. But it looks of it, yeah, no, here we go. Cytosol, we're good. We do not need the organelle. So by the looks of it, some of this is done in organelles. So, like, some of the precursors are made in the plastid or mitochondria. But the actual capsacin seems to just be made in the cytosol, so which is just like the main goopy compartment of a cell. So yeah, cool. This gene itself doesn't, but the other one might. It was, what was it? It was 𝐵-cat. Is that the? Where'd it go? 𝐵-cat. Okay, branch chain aminotransferase. Let's look this up so we know how it works, because this is the next thing that we're going to need. And we're going to use the P2A system again, because I don't want to stick another promoter in here. I don't see any point. But we'll see if this needs a localization signal. I'm looking up for 𝐵-cat. I don't know if 𝐵-cat needs it in an organelle. To Wikipedia, yay! Okay, let's plants. Useful in scent and flavor. This does not tell me where. I'm gonna keep an eye on the comments. Does anyone know if this thing is like? Can somebody check and see if this is actually supposed to be in an organelle or not? I need to know what this actually does, so that I can see it. Okay, branch chain amino keto acids. Presumably this is the thing, and so it makes that okay. Interesting. Okay, so you've got. So what goes in? So if you got valine, you get this. If you get isoleucine, you get this. Okay, so this works on a variety of different things. Let's go back to that. So if we know that this is working on the leucine, or like if this is working on amino acids, basically, then I should be able to see where that was in the pathway. Yeah, I think it might be in mitochondria. What am I looking at? You have a variant for the mito and chloroplast. Produced in the mitochondria. Okay. Bugger. So that means I need a mitochondria localization site, which is not something that we've done on this show before, so that's exciting. Come on. Cooperate, please. Produced in mitochondria. Okay, I'm seeing that. Thank you. So now we gotta find a target peptide. Although if it's a peptide, that actually makes my life easier, because it's not like some weird promoter nonsense. So it should just be able to be made with P2A. Let's see what we got. Wait, you know what? Can somebody find me an 𝐎𝐫𝐢𝐠𝐢𝐴𝐺 plasmid with this thing in it? I'll keep looking, see if I can find a name, but I'm sure that y'all know more than I do. So, if somebody can find that, that would make my life a lot easier. And I'm just gonna keep an eye on the chat while I look this up. Man, I've had requests to do the stream later, earlier, all the time. I'm doing it when it's convenient for me. Sorry. Like, y'all live in different places. can't really do anything about that. Some of you are in Australia. It's like 5 in the morning there right now. I'd love it if everybody could all tune in at the same time, but this is the problem with living on a globe. Okay, thank you, Ron. You the best. I'm going to keep looking this up. This says 𝐎𝐵𝐾, so that is human, which is useless to me. Well, I mean, it might be the same. I don't know if it matters, but, like... I very much would love to put something into space. That would be great. Ideally myself, but, I mean... I don't know if I'll ever go to space. But definitely a satellite. I mean, my 5-year plan includes putting a satellite in orbit. I had some friends who were starting a rocket company to try and do that, but the rocket sank, so I don't see that happening anytime soon. So it is just a transit peptide. Well, there's a signal peptide and transit peptide. Cool. Very cool. But how about a sequence? A sequence handy? Oh, see. Oh, you know what? This is a perfect example of the thing I was talking about earlier. Where somebody included the sequence, but they did it as an image because they're bastards. If you didn't add the mitochondria localization gene, it'll just try and run in the cytosol, which sometimes... So the reason that you've got to localize to, say, the mitochondria or plastids or whatever is because the reaction conditions and the things that those organelles can survive differ. Okay, thank you, Ron. Is that the... fuck, how am I going to copy this? Did that switch my... no, wrong thing. Sorry. I meant to do this. My bad. So, yeah, I'm just going to do this so I can pop over to the chat. Stop moving. Okay, cool. Ron, what is this? It's the mitochondrial localization. Is it a transit peptide too? Does it do all the things? Or, like, what's the ruckus? If I hit... Yes, good. Wonderful. Okay, cool. Okay, so I'm going to just... Where can I put this? All the usual places where I would post things is not a place that I really want to open up on live. Yeah, seriously. Yeah, just pasting DNA in the chat is the reason I love this show. Like, this is so much fun. Okay, so I'm gonna paste this in here, just so I don't lose it, and I'll make an annotation so I know what it is. Mito peptide. So I'm gonna have to add. Well, I was gonna do P2A. That was the thing. Come on, work. 79135. Okay. So I'm gonna do the same thing. Okay. So, oh, wait, we got a message from Ron. Must pay attention. Okay, so Ron says it's the same thing. The peptide is removed after the transport. It is part of the main protein and is cut off after transfer. It needs to be in frame with the protein. I mean, it is in frame with the protein. ATG, we're good. As long as you didn't miss a letter. But, I mean, I can just do a little create translation so we can make sure. Okay, cool. Now we can see all our aminos. Okay, does P2A work in plants? I think it does. P2A peptide. And so we used this one in the first one, in the first stream. Why am I doing this? I have this literally as I said it. I'm like, wait a second, we have this. Milk and eggs. Let's grab the milk. What can be edited? Okay, cool. No, this is great. Thank you. So yeah, this is the milk and egg plasmid. Oh, by the way, so I mentioned that I made some changes to this for the new upload. Close this, just so I can make this a little bigger so I can actually show you what I'm talking about. Okay, so this was the thing that we made in the very first stream. It's the this is the milk plasmid that we're gonna have synthesized. You'll notice that these little triangles here are now different colors. These are the P2A peptides. So the way P2A works, they're unfortunately they're called self-cleaving peptides, but that's not actually how they work. The idea is you copy this sequence and you paste it between two different proteins that you want to have made off the same promoter, which I'll show that in a second. Or actually, I can just show it here. Is it gonna? Of course, it's gonna. Wow, just really zoomed in way further than I wanted. Okay, here we go. So this was the thing that we made in the first episode. So this is the promoter. This is like this is where the enzymes bind to turn this DNA into RNA. So this pink and this blue green is the first protein that we wanted to have made, but then we wanted basically to make more proteins off this one promoter instead of having to add a new promoter every single time. So what we did was we added what's called a 2A peptide. It's self-cleaving. It's not really how it works. It's more like a record skipping. So it makes this, so once this is RNA, a ribosome will start copying all of this and turning it into protein. When it hits this part, it basically like skips. So it ejects the protein that it was working on and then like skips and then starts working on the next one. So you can get multiple proteins out of the same single RNA. So it's efficient and it's easier to synthesize basically. So originally we had 3 of the identical, there's multiple different ones of these. We just used P2A every single time, but the change I made is I went in and I switched 2 of them out for T2A. So there's some variation just because if you use the same one every single time, it can have issues. So we've got P2A, and I'm going to use P2A just because I'm feeling it. And give that a little copy, and go back over to our 𝐾𝐵𝐈𝐴 over here. Cool. And so that's going to get pasted in right here. Cool. So now it's going to do the capsaicin. So if we look at this, it's going to. So it starts with this promoter, which Ron provided, and then it's going to do capsaicin synthase. I can't seem to say synthase today. Synthase, which is the last step in the spicy tomato pathway. It's one of the 2 genes that are missing. And then we do P2A, so it's going to eject this and then start working on the next one. We've got our mitochondria localization peptide, which Ron, the frickin' mad lad, just posted the code in the chat like a savage, but I appreciate it. And so now we've got to add the last gene and then a terminator, and then we, I think, are good. Okie dokie. Man, plants are weird. Okay, so we need this branch chain something something thing. BCAT. Okay, BCAT. See what we got. Branch chain amino acid aminotransferase. I'm going to grab the plant one because that seems effective. Okay, that makes sense. Man, there's an experiment that I've wanted to do for a really long time. So there's this thing called, and I brace for a tangent, boys, because this is this is something I've been thinking about for a long time. But so there's this thing called cell-free protein synthesis, which is really weird. You basically get a bunch of an organism, usually yeast or E. coli, and you basically explode them with ultrasound. But you do this just gently enough that it pops them all open, but it doesn't damage any of their internal um, like mechanisms. Like all the proteins remain intact. And then what you can do is you provide all of the amino acids, all of the nucleotides, all of the stuff that you would need to basically do DNA synthesis, RNA synthesis, and protein synthesis, and then some fuel. So like some like ATP, creatine phosphate, this sort of thing. So basically, you need to provide all of the things that an organism would need on like a molecular level to function, but it's dead. So you can basically make it make whatever protein you want, because if it's toxic, it doesn't matter. Like if it's something that would normally kill the host organism, who cares? It's already dead. It's basically like a tube of like zombie E. coli. So it's like one giant E. coli that's like 5 or 10 milliliters. And you can just have it running and make you whatever you want. And so people use this for assays and like making stuff. I just think it's really cool. But the one of the things I wanted to do was like, how could you do this on the minimum budget? Because the reagents are expensive. I'm gonna see if I can look up some pictures so you guys have something to look at while I ramble. But I was basically thinking like, yeah, this is the idea. So I'm thinking about like, how do you do this for the absolute bare minimum amount of money? And so I was like, okay, well, what if you start with some yeast, like some baker's yeast? Because baker's yeast is great because it's like just pure, it's basically just like pure cells. It's like gram quantities of cells ready to go. So, you grab some baker's yeast, pop them all open, so you've got your extract. So you'd have like this part, right? So you'd have your cell extract, which has all the like machinery in it. But then it was like, okay, but how do you provide like all this stuff, the like salts, the cofactors, amino acids, like DNTPs, like how do you get all that? I'm like, wait a second, what if you just took another batch of yeast and just autoclaved it? So just like boil it basically. So then you'd have essentially all of this stuff that you could possibly need. And I mean, the other option is you could add some like protease to it first. So like some pepsin or trypsin or something. So like any proteins that are in there would get broken down to amino acids. And then you boil it so that it turns anything off. For ATP regeneration, I mean, like you could provide some ATP and some creatine phosphate. But I'm like, there's one. So the thing that I actually want to test is, what if you just added mitochondria? Like if you extracted mitochondria from something and just added it directly to this extract. Because if there's one thing that I learned in all of my bio classes, it's mitochondria are the powerhouse of the cell. And it's where a lot of the like ATP regeneration happens. So I was like, what if you just added some mitochondria? Like would that work? And so then you could just like give it some DNA and see if it runs. Yeah, so that sounds really fun. I really want to try that at some point, and I'm locked in my house, so I just might. And I already have all the ingredients. And if it's the only thing that I ever learned from, if it's the only thing that I learned from school, yeah, I know you can extract mitochondria from cauliflower. That's totally what I want to do. I want to like extract some mitochondria from I was thinking spinach or cauliflower or whatever, and then just do 2 yeast extracts, one unbroken, one boiled. Then throw in a little ATP and some DNA and just see if it runs. Just because that sounds like if it works, I mean, it's so stupid, but like it'd be so funny. And also it'd be cheap. Like it would cost nothing. Like ATP costs nothing. You can buy it on eBay. I have a bag of ATP And like when I bought it, I was like, what would I even do? Like, could you eat ATP? I was like, no, you really can't. ATP is fuel when it's in a cell. It's like a signal when it's out of the cell. So you'd basically turn to mush. It's no good. But it's funny to own. What's the line between lysis and hosing the contents of the cell? Temperature mostly, and also ultrasound power. Like if you're just chooching like a kilowatt of ultrasound in there for long periods of time, like you're gonna break all the proteins. But if you're using like 40 watts of ultrasound and it's real gentle, you can do it pretty easily because then like you can just pop everything open, spin it down to get the cell wall out, and you should just have a really nice protein extract. There's also something that's really interesting is basically like the shit. I lost my train of thought. Fuck. I had such a good thought and then it just gone. Well, I guess the Alzheimer's is setting in. Oh no. Shit. What was I gonna say? I have no idea. But I've got a friend who's working. She wants to use, for all the bazillion people asking about COVID. She wants to use this sort of thing, like this sort of system. Ow, 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 ow. Leg cramp. I bent my leg funny, and it was like, fire all cylinders. Ow. Anyway, so I've got a friend who wants to use this cell-free extract to do viral testing. So the idea is that you could like add your sample to it, and it would change color, like using some really clever ribo switches. So basically, like RNA that can like target sequences. It's really clever. The idea being, excuse me, that the ribo switch would like change shape, and if it does, it lets it lets a gene be synthesized, which would make the solution change color. So it'd be like a colorimetric thing. Although I actually want to talk about colorimetric COVID tests. I mean, you're going to see a video about it soon. But yeah. Anyway, so massive distraction aside. Okay. Okay, cool. And this one does valine. This is exactly what we need, I think. Let me just check. Yeah, so you need valine to 𝐎𝐫𝐢𝐠𝐢𝐳-CoA. So I'm going to just keep this one in mind. Okay, now I'm gonna stop being distracted now. So if anyone can find me the right branch chain amino acid aminotransferase for this, because I don't know if there's a difference. I understand that there's like a whole family of them. So yeah, if anybody can find these from, what's the name of this species? Just so I know. Hot pepper. Okay, well, that's not helpful. Let's just add pepper on there and see what happens. I don't know if it matters. We definitely don't want the, genome sequence of hot pepper. That is exactly what I want. Yeah, liquid ozone is spicy. Go watch Explosions in Fire if you want to see liquid ozone. That stuff is terrifying. Capsicum. I should have just put capsicum. Capsicum atom. Okay. Let's see if it's on UniProt. not seeing... I'm not seeing the thing I want. Can we get some help in the chat here, please? I know there's many different species of pepper. I'm looking for, this is human. That's not helpful. 𝐵𝐶at. Is it 𝐵𝐶at 1? I don't know if it's going to be different. Hide. Why are y'all talking about rockets? I need help with plant stuff. Can y'all just like chill, please? I'm looking for this branched chain amino acid aminotransferase from peppers, ideally. Rockets are cool, but that's not... Oh, what is this? What happened here? I just pasted in... Oh, because it had 𝐵𝐶at. Okay, that makes sense. Rockets are amazing. It's also not the topic for today. So, I need help with this. So, if y'all can chill the fuck out for like 4 seconds, that'd be great. So, I'd really, really like this. You know, okay, well, there's 50,000 of these things to go for. Well, thank you, Jesus. Okay, we're looking for a 0. Is that a D? No, that's a 0. Top. Fuck. Okay. 2GX 9. Did I type that in wrong? I feel like I typed that in wrong. AOA 2G 2X. Oh, that's supposed to be a 0. It's also supposed to be a 0, I think. I did. Yay! That'll do. Alien. I mean, this looks like it does the same thing. 𝐎𝐵ROT. Q9. Fuck. 4CF8. I'm just looking a couple of these up because I want to see if there's any difference. 4CF8. Yeah, this looks right. Capsacin atom, branched chain amino acid aminotransferase. LV. Yeah, I mean these all seem like they do exactly the same thing. Like the catalytic activity that's listed seems to do the thing, so I'm going with it. We're saying it's good. But this one is Capsacin atom, so this one is from the exact species that we want. So whoever found this one, I think this was Gustav. Thank you very much. Q94CF8. Yes. I know. I'm there. We got it. Thank you very much. Coolest of legumes. So we're going to do the same little flipperoo thingamadooey that we did last time. Reverse translate. Still on one line, which is good. We're going to pop back over to here. Let's create that annotation. This is 𝐵𝐶at from C anim. Oh, 2 U's. Knew that looked too short. And we're gonna make this a different color. Let's go with yellow, I think. I don't know. No, let's pick a different color. Let's go blue. Save. Wunderbar. Alright. Let's do a little translation-y goodness. Yeah, it's all in frame. We're good. So, yeah, I think that's actually... Oh, Terminator. It's actually, this is really almost synthesizable. Okay, cool. So now we've got all the stuff. So the last thing that we need is the Terminator, which we loaded in at one point. Was that this? Is that what this was? No, this is the promoter. What did we do with that? I have to go back over to the spicy tomato. Did I not load it in? I downloaded it. So I have a really interesting pepper growing right now. It's called a 𝐎𝐫𝐢𝐿𝐴. I'll show you a picture of it because it's, I mean, I don't have any done yet, but like it's sprouting. If you've not heard of these, I'd highly suggest them. I mean, I say that I haven't actually tried one yet, but it's a habanero that's been bred to have no spicy. So it's the exact opposite of the thing that we're trying to do today. So like today we're trying to make a spicy tomato. This is a habanero with no spicy. So it's all the flavor of a habanero with none of the spicy. And so I've got these growing. I've got two of these little plants growing. Highly suggest trying them out. I've heard very good things. Also, this is the site that I bought them from. Also a site I would very highly recommend. This place is awesome. The variety of seeds that they carry is really, really cool, and really, really cheap. They send you free stuff as well. They're fantastic. This is not sponsored, by the way. I just really like them, although that would be such a cool sponsorship. Yeah, somebody ended up actually making a sorbet out of habanada because they're so sweet and tasty, and apparently it like wowed a bunch of chefs when it was first unveiled and something. I don't know. It sounded pretty good. Cool. So I've got the Terminator. This is the bit we're gonna have to stick on the end, and then I think we are done. Let's just jump to here. Okay. So we need to tell this to stop now. That's put it on the table. So this is the one last thing that we got to do, and this is something that I'd forgotten in the first time. So let's grab some stop codons. So these are the stop codons. We've got TAA, TAG, and TGA. So I'm gonna just throw one of each in, just because I like to make sure. I want to make sure it's it's really stopped. Did it? It did not. Okay. TAG, TAA, TGA. So yeah, I throw one of each in because I mean it costs nothing, and it guarantees that it stops. So yeah, you can see them here. They're little stars. Just tell you that it's like off. We're gonna add an annotation just because I like to make sure that people know what this is. Stop codon. And then the last thing we're going to do is we're going to paste in that terminator that we got from the other thing. And then I think we're done. What time is it? It's only like 3:30. That one was quick. I'm gonna have a little look-see at the comments, see what's going on. Cool. Let's close this. Make this a little bigger so you can see. Oh, too big. It's gonna freak out when I do that. Yes, it is. Just a wee bit smaller. I wish I could reposition this. That's really kind of annoying. Okay, well, I think this is actually theoretically done. Theoretically. So let's review what we have done. I'm just seeing. Okay, that is the second shit post I've seen from you. You're banned. Okay, so let's do a little review of what we've made. So I'm gonna just click this. Oh, also I'd like to make a note. Projects, notebook. There's no spicy tomato notes. So I'm just going to add a blank entry. So 𝐴𝐵𝐷𝖆 2300 was cut with 𝐵AM_HI and insert the spicy jeans. Cool, just so I have that. Just like to make sure I've got notes, because if we ever actually wanted to make, because I mean, at this point we could actually make this pretty easily. Cool, so let's just do a little review of what we've made. So we started with 𝐴𝐵𝐷𝖆, which I might actually have open, and I think I do. Yes, I do. Awesome. So we started with this. Oh, Jesus. Yeah, we started with this. It's 𝐴𝐵𝐷𝖆 2300. So this is a piece of DNA that is designed to be able to be put into a type of bacteria called Agrobacterium. That bacteria is very good at adding DNA to whatever plant you expose it to. So what you could do is you could take this DNA, modify the agrobacterium, use the agrobacterium to modify your plant. Cool. So then what we did was we cut it open. We added the CAMV35S promoter. So that's cauliflower mosaic virus 35S promoter. Actually, we added it here. That's the other one. So we added this one right here. So we've got so this will just have this gene run basically anywhere that, like, throughout the entire plant, basically. Then the first gene that we added was capsaicin synthase. So this is the last step in the capsaicin biosynthesis pathway. Then if we scroll down, we've got P2A. So this is what's called a self-cleavable linker. So that basically this means that we've got the one promoter here. It'll make this first gene. It'll hit this P2A, and then it'll basically eject this and then start making whatever comes after it. Which in this case, first we've got a mitochondrial localization peptide. So this basically tells the plant, put whatever this is stuck to into mitochondria. And then it'll do that, and this apparently gets just cut off as in that process. Then to that we added this gene. It was BCAT from a spicy pepper species. This is a branched chain amino acid aminotransferase. It's one of the steps in the pathway. We'll look at the pathway again in a second. And yeah, so then the last thing that we did was we added a stop codon or 3 and then a terminator, which basically marks the end of the gene. So in theory, this whole thing, I wonder if I can get rid of this annotation. In theory, you could put this into tomatoes and it would make a spicy tomato. So I'm just going to get rid of that because I'm sick of looking at it and it's not really necessary. Cool. So that is actually the thing. How large is this? Just because I'm curious. To here. Length 2791. So that's actually not bad. That's about a $700 plasmid. Why you didn't add P2A in the penicillin project? Because the penicillin project, each one already came with a promoter, and I wasn't actually thinking of synthesizing it. Whereas this one, I'm almost tempted. I don't think I will, just because I don't have agrobacterium. I don't work with agrobacterium, and I don't really do anything with plants. I'm starting to do stuff with plants because I'm interested, and it's weird. But all of my plant work got destroyed when the pandemic hit because it was all at the lab, and so it didn't get tended to when it needed to, so I've got to start it all again, which is annoying. But yeah, so in theory, you could take this and you could put it into a tomato, and it would, in theory, make a spicy tomato. The one issue, and this is something which we can maybe now look at because it's still pretty early, we've got some time. This is not what I want. I wanted this. Okay, so we've now basically fixed this pathway. So you'll see that BCAT and CS were the ones that were missing. These are all the different genes that you need to make a spicy tomato. And you'll see that in a sweet pepper, you've got most of the genes, and it's doing good, except the very last one that actually does the make spicy is missing. In a tomato, same basic idea. You've got most of them. The levels aren't super high, but you're like just straight up missing 2 of them. And this could just be a matter of the promoter is not sufficiently strong, or doesn't run in the fruit, or there's a bunch of different reasons that it could happen. But point is, it was missing these 2 genes. So we made a piece of DNA that will replace these 2 genes because it's very easy to just add genes comparative to like messing with promoters that are already in there. Yeah, so in theory that should fix this. What you could do is you would want to check, and I mean this is getting into the, like, you know, if you were doing this as, you know, your thesis or whatever, rather than just like fucking around and doing it for fun. But if you really wanted, what you could do is find a variety of tomatoes. So like do a whole bunch of like sequencing stuff, or maybe HPLC stuff if you could find a way to tag these. Maybe like an antibody-based test or something. I don't know. But if you could figure out whatever, I mean, in theory, this paper would explain how they did it. But like, if you could find a variety of tomato that had higher, just inherently higher levels of these, and then you added that plasmid, you should get a fairly spicy tomato. It seems like these 3 also need to be pretty high levels. So in this case, the thing that we made, we're going to end up with a very high level of the capsaicin synthase, but fairly low levels of the other things. So the amount that you're making is probably not very high. So it wouldn't be, it'd probably be like a banana pepper, basically, but it'd be a tomato, which is, so, I don't know. It sounds like this could be very cool. Also, there's a bit of a, like the way that we'd actually get, so there've been many questions in both this stream and other streams, about, yeah, BCAT would also probably be fairly high. But so there've been some questions about, like, how do you actually get this DNA? Like, once you've made it, like, how do you get this into the thing that you want to express it, basically. So in this case, we've already kind of talked about that. You'd use agrobacterium. So you would take a little piece of tomato leaf, and then you would put it on a special type of media, which makes it turn into like this little plant booger. You can also just germinate the seeds. So plant sprout. Let's see if this comes up. So the idea, so this is actually something that a friend of mine has been messing with, where rather than doing this process of, you know, start with a leaf and then grow it into a little plant bugger and then modify it and then grow from there. What you can do is you basically sprout the seed. So just like the tiniest little bit of sprout is sticking out. So you might be able to see that on this one. Yeah, so you'd basically do this, excuse me, where you just sprout the seed just the tiniest little bit. And then you would treat the seed with agrobacterium. So you don't need to mess with any sort of plant culture. You just basically soak the slightly germinated seeds in the agrobacterium, and then you do your antibiotic wash to kill off the agrobacterium after a day. And in theory, that should just sprout into a modified plant. So that's the easiest way to do this by far. So yeah, I mean, that's how you would do this. It's how I would do this. The other option, if you don't want to do any of that, and more to the point, if you would actually want to, because if you actually put this into a tomato, it would immediately be illegal to sell. I'm about to talk about gene guns, but so if you were to do this using agrobacterium, it becomes immediately illegal to sell, at least in the U.S. The reason being that in the U.S., the only plants that are genetically modified that are allowed to be sold. Oh, thank you, Connor. Yeah, so the only plants that are genetically modified that are allowed to be sold can't be modified with agrobacterium, because it's mostly for stupid reasons that you're supposed to or there's a slight risk that if a wild agrobacterium were to infect that plant, it could then pass that gene onto a plant that you haven't registered, kind of thing. So they worry about like the propagation of the thing, which personally I find is just fucking stupid. But whatever, I don't run the EPA. But the so if you wanted to actually sell this, like if you wanted to make this into a form that is sellable and like something that you could share, you would have to use what's called a gene gun. So let me show you what a gene gun looks like, because gene guns are dope. This is a gene gun. It looks exactly as amazing as you'd think. And it literally does exactly what it sounds like. It shoots the DNA into the thing. So the way this works, let's see if I can find you a picture that isn't just potato tiny. So basically the way this works is you've got like a burst seal right here. So this would be pressurized. There's a burst seal here, and then a little, usually it's a piece of like parafilm, or I mean sometimes it comes with special burst discs, but I mean parafilm works just fine. And you basically put little particles of either teeny tiny particles of tungsten or gold that you've soaked in the DNA solution that you want to transform into the plant. Or, I mean, technically you can do this with almost anything. But so you soak your DNA or your gold particles in DNA, you dry it onto this little burst disc, and then when you fire the gene gun, it sprays these particles at the cells very, 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 very fast. And here's a picture of what this looks like. I mean, you're literally like just shooting DNA into things. It's awesome. So when you do this, that little area would end up, some of the cells would end up taking up the DNA. So the little particle literally like just puts particles into the cells. Some of the cells will die inherently. You're shooting them with a gun. Some of the particles will end up inside of the cell. The cell will patch itself back up, and when it does this, the DNA ends up getting inside of the nucleus and then starting to run. What you can then do is you would take that little section of leaf that you shot, and you would put it on a plate to grow the little plant booger. But you would also include an antibiotic, usually hygromycin. It could also be kanamycin. There's a variety, or 𝐲418, or a variety of other things. Depends how you designed your DNA. And so what this would do is it would kill off any cells that weren't modified, like that didn't take up the DNA, and so the cells that survive would then go on to form stem cells, and then you could induce them to basically turn into a whole new plant, and then that whole new plant, every single cell would have the new DNA. So in this case, you could have this made, and then load it onto the little particles, and then load the particles into a gene gun, and then shoot a plant with it, and then culture them. And in theory, you would end up with a spicy tomato, if you did it to a tomato. If you did it to something else, you may end up with a spicy something else. But then you'd have to add a whole bunch more genes. And so this is if we wanted to do this in yeast, we would actually have to add a bunch of other genes. So like we'd have to add all 9 of them, I think. So we've only got 2 here. So if we wanted to do this in yeast, we'd end up having to do another like 7 genes. Could you shoot a person? Yeah, you could like this works on almost anything. It's a little harder with bacteria just because they're so tiny, but if you had small enough gold particles, you totally can do that. I know you can do it with yeast, and now that I think about it, I'm beginning to contemplate whether or not I want to try the spider silk thing by gene gunning it into the yeast, because evidently trying to heat shock it in doesn't seem to be working. The spicy skin challenge. Yeah, please don't do that. It might also work on a potato, but the problem with the potato is potato fruit are poisonous. Like people, it's funny, the whole Solanaceae family, like if you eat anything other than the fruit or just the fruit in many of the things in that many of the plants in Solanaceae are just very poisonous. I mean, deadly nightshade, for example, is totally in the Solanaceae family. Like they're all the nightshade family. So in theory, you could, but it's I don't know if you'd end up having to add like a tuber localization thing, and you'd realistically, like if you wanted to do this, if you wanted to make like spicy potatoes, you'd probably end up having to add a root or tuber selective promoter, and you'd end up having to include all the genes. Like I doubt that these run in the tubers. They probably only run in the fruit for the most part. A full kit. Oh yeah. So buying a gene gun is insane. Don't buy a gene gun. It's fucking compressed air. So actually, over on the Sci-Hub, or not Sci-Hub, the Sci-House YouTube channel, there's a video on how to make a gene gun. I'm probably going to redo that on my own channel, because thank you, Sans de la I can't pronounce your name. Thank you, Sans. Would it be possible to genetically engineer something to excrete psilocybin? Yes, it's been done in yeast. But yeah, so it's totally possible to build a gene gun from like hardware store parts, and you can buy these little things. They're like for like basically like an emergency bike tire inflator that runs off little compressed CO2 cartridges. So you can literally, using like hardware store parts and one of those, you can cobble it together, and like a solenoid valve, you can totally make a gene gun. I've done it. It works really, really well. You can buy the tungsten particles off of Amazon, and it works great. So I'm actually thinking about, now that I mentioned this, I think I'm going to build a gene gun. I mean, it doesn't cost very much. Like, I think it cost us $100 in parts, tops. So yeah, I think I'm going to go ahead and build a gene gun. I'm not going to gene gun myself, because I'm not insane. That's not true. I'm I'm not insane enough to shoot myself with a gene gun. Although, maybe. Anyway, I think I'll, I think I'm going to build a gene gun and either try and use it on plants or maybe some yeast. I don't think I'll do it on any kind of mammalian cells, but yeah, that could be fun. No, the only reason I was thinking of gene gunning a person is you could use it for like vaccine delivery, which now that I think about it, would technically be a easier method than electroporation. It would probably hurt less, or would it hurt more? I don't know. Unsure. Kind of the issue is with gene guns specifically is the keratin layer on the outside of your skin is really hard to punch through. So you either end up having to use a lot of pressure, which just kills more cells, or might have to like cut yourself open first. I don't know. I'm not familiar with in vivo gene gunning. I'm not sure that's even a thing. So I'd have to look that up. Anyway, so yeah, I think we're basically done. I mean, it's been 2 hours. Our plasmid is made. I'm gonna upload this to the GitHub so that anyone who wants can play with it. And this is your reminder. Please go over to the GitHub if you're biologically versed. Check out the milk and eggs plasmid. These are the new versions. I just uploaded them yesterday or today. It was 17 hours ago. So yeah, I did it last night. These are the new versions. Please have a look at them. Check and make sure that I didn't miss anything or there's nothing stupid. And then basically Monday, I'm going to be putting in for a quote request to actually get these made, because we did a fundraiser and I want to have them made. I think they're really cool. So we're going to get that done. Okie dokie. Yes, I'm seeing the request for a Discord. I see them. I'll think about it. I don't generally do Discords, but we'll see. So yeah, milk and eggs, please check that. This is going to get uploaded. I'm going to do 5 or 10 minutes of question time, and then we're going to wrap it up for the day, because we seem to have accomplished the thing. I don't know. I think I might add this one as a, if people vote for it, we might do a fundraiser for it down the line. But it's not something that like I am really super passionate about. I think it's funny. But I don't know. Okay, so do we have any questions to end out the stream for the day? Other than, can you make a Discord? What happened to the plastic eating mushrooms? Turns out they make poison, so we stopped working with them. Can this same plasmid be used on a gene gun? Yeah, it should be. Overall, how many of the human body cells are reachable as targets for genetic engineering? I'd say almost all of them. It just depends how you're delivering the DNA, mostly. If you're using viruses, like, you can hit most tissues. Even the brain, honestly. There's a there's a treatment for prion disease that involves injecting, I don't know if it's just the antibodies or if it's DNA as well, but yeah, if you've got like a prion in your brain, they can basically stick your brain full of frickin' antibodies and it fixes it. What are your thoughts on which fuel I should use? Should ethanol? Is this for rockets? If it's for rockets, then what color are the milk and eggs? They should be colorless, ideally. Can you recommend a learning path from learning to application of this? Thing number one, and I've said this in every single stream, and I'll keep saying it until everybody does it. Buy yourself a copy of the manga guide to molecular biology. From there, you know, get a little lab set up, pick a project, and just work your way through it. If you were not lactose intolerant, what would you have done? I don't know, something else. Kevin asks, I have $1,000 coming to me and I want to set up a lab for making yeast-produced CBD. What equipment I would need? Well, you're going to need some piece of equipment that's going to add about 4 zeros to that number, because good luck with that. I have a friend who's done it and it's cost him 10 million. So good luck. When I was thinking of gene gunning yourself, what did you have in mind other than vaccines? Nothing. I was thinking of vaccines. Oh god. Shit. It scrolled too far. Y'all, some of you are about to get fucking banned if you keep asking about the lactose intolerance. Like, this is really getting obnoxious. If you were not lactose intolerant, what would you have done instead of what you have gone into now? Probably the same. The reason in the video about the lactose intolerance, I said it was because of the lactose, like I went into bio because of lactose intolerance, but that's not actually strictly true. I'd always been interested in bio, and the actual reason that I went into biology is because I watched a Studio Ghibli movie called 𝐎𝐵𝐺 in the Valley of the Wind, which is a fucking phenomenal movie, and I highly suggest it. I literally made it like halfway through that movie, and I like paused it, went upstairs, and submitted my application to like a bio program. So yeah, that's why I went into bio. I was stuck deciding between nuclear physics, genetic engineering, and theater. And I watched that movie, and I was like, okay, bio. What else we got here? I don't know if I'm going to do the tomato project. I'm going to this is one of the first genes that we've made lately, or plasmids. I mean, again, I'm going to be doing these episodes Mondays and Fridays for the foreseeable future. So we're going to end up with a lot of plasmids that get made. So I'm probably going to do another like 3 or 4, maybe, or 6. I don't know. We'll do a few more, and then if we end up with a collection of a few that seem viable, I'll put it up to a vote. Unless one really stands out as like, yes, this is amazing and we need to make it, I'll probably put it up to a vote to see what we synthesize next. And so yeah, if we happen to do the Capsacin thing, then the plasmid will be on GitHub for review. Oh shit. Oh god. It keeps jumping. I'm trying to keep up on all the questions, but y'all are sending like a bazillion at once, and I'm still fairly far behind. Okay, that's okay, fine. Scrolling. Make a robot that helps you in the lab. I have a robot that helps me in the lab. It's just at my other lab at the moment. I've got to move it here. Can you make a cellulose digesting E. coli? Well, we just did a cellulose digesting yeast. That was what we did on the last stream. If your choices are between pure hydrogen peroxide and ethanol, you'll probably get more bang for your buck with ethanol, but it depends what sort of rocket you're making. If you're messing with constant like pure hydrogen peroxide, I mean, first of all, it's just a nasty compound to work with. But I mean, liquid oxygen isn't any better. Rockets are just very dangerous. I don't know, man. I haven't made enough rockets to be able to give you a clearer answer. How do the genes get into the gold particles? They don't get into the gold particles, they stick to the gold particles. So you literally get gold particles and then you just like soak them in the DNA and the DNA just kind of sticks to them. DNA is sort of a sticky molecule, and you like adjust the the stuff that's in that solution to make it preferentially stick to the gold. Can you repeat the movie name? I want to watch that. It's called 𝐎𝐫𝐢𝐠𝐢𝐳 in the Valley of the Wind. It's made by Studio Ghibli, spelled 𝐔𝐩𝐈𝐵LI. Fuck it. This movie. This movie is the reason I went into biology. I cannot recommend it highly enough. It is just so fantastic. Go watch it. There's a scene about halfway through, and for those of you who've seen it, it's the rock trees. The rock trees were the reason that I went into bio. How do you get the DNA off the gold? The DNA once it's in the cell, it lets go. It's a Ghibli movie. Of course, it's fantastic. Duh. If any of you are not into Ghibli movies yet, just because you haven't seen them, fucking get on it. They're the best. Like I'm a huge movie nerd. Like I grew up around movie stuff. I very much love animated movies, especially, and there are very few that are on par with anything that's well, not anything. We don't talk about Tales from Earthsea, because fuck that movie. But the rest of them are so good. If I had to like pick, what? Yeah, just not Earthsea. Isn't it Japanese? And I mean, for those of you who are plebs, there is a dubbed version, but just watch subtitles like a normal person. I didn't like Mononoke, and I need to rewatch it because I get hate for this every single time. But just it wasn't my jam. Like, it was very pretty, but it's just, of all of the Ghibli movies, like Nausicaa, Porco Rosso, and The Wind Rises, far and away my favorite movies. By a mile. Oh, Howl's Moving Castle, also fantastic. I don't know how we got on this, but freaking go watch these movies. They're so damn good. Wait, what? What are your thoughts on anti-GMO protests? I think they're dumb as fuck. You're asking a genetic engineer what I think about anti-GMO protests. I think they should kindly shove their incompetence up their ass. You can send me pics on Twitter. Yeah, Ponyo is pretty cute. I mean, again, there's hardly a bad Ghibli movie. They're Ghibli movies. Do you have any degrees? No. I started doing university, realized it sucked, and then I stopped. Could something go wrong with GMO due to depletion of some precursor or something like that? Probably not. The thing would just die. So this is actually, this brings up an important point that I feel like talking about. So GMOs, highly regulated. Everybody loses their fucking minds when you release one. What's not highly regulated is what's called nuclear gardening, which is basically you irradiate the ever-loving fuck out of the plant, and then until you get one that has a weird property that you like, and then you just like breed that into your usual stock of plants, and you just hope that it makes the right thing. The ruby red grapefruit, that's red grapefruit. If you've ever had one of these, that's how that was made. They irradiated the ever-loving hell out of a grapefruit and waited till it turned red, basically. I'm mostly hyperbole, but that's not really. Like, that's what they did. Like, they made a weird grapefruit, and they bred it with the other grapefruit to make a weird red grapefruit. They did this with a cesium pill. So it's basically... let's see if I can find you a picture of this. Yeah, so it's basically this. So what you do, is this a good picture? Yeah. So basically what you do is you get this like big circular field where you put all your plants, and then right in the middle, usually, you have like a pedestal they'll raise and lower out of the ground. And inside is just a hunk of like cobalt-60 or cesium-137 or just something that's very, very radioactive. You basically raise it out of the ground, expose the plants in the area to it, and then you lower it back into the ground so that it's safe to go into the area. And then you just like take the seeds from those irradiated plants and then grow them and see what happens. This, totally not regulated. You can do this. No extra approval. You're messing with thousands of genes at once, and nobody cares. But you mess with one gene, like just one, and everybody loses their fucking minds. It doesn't make any sense to me. Taste Emporium update. The next Taste Emporium video should be out in 2 days or 3 days, depending on how long it takes me to edit it. That's what I'm doing for the rest of my day. We're going to be making some bread. Styrofoam worm project update. The update is there's no update, and there probably never will be. We tried it, didn't work, moving on. What else? Did I miss any good questions? What are the most important concepts in chem for bio to really grasp? Since bio is just weird chem, and I haven't had chem for the last few years. Depends what sort of bio you're doing, and it depends exactly what you're doing. This is one of those, the more chem you can fit into your brain, the better, but also biochem versus ochem overlap, but are not the same thing. Like, different reactions can happen in bio that are just unfeasible to do in a beaker. So you basically pick up a biochem, like I'd pick up the manga guide to biochemistry is a really good way to start if you want to really dig into that. Yeah, exactly. It's the laws around genetic modification are insane and stupid, and I hate them. It makes no fucking sense. I mean, but I'm also of the opinion that the world is already fucked. So like, this concept of like preserving the ecosystems is already like, it doesn't make any sense. Like, we've lost like 90% of all species, of all macro species at the very least. We're also losing like most of the insect species and plant species and everything else. So like, I past a point, I'm all for releasing some new biodiversity and just like giving everything a good mix and seeing where the cards fall, because like everything's fucked. Who cares? Obviously, I understand that like conservation is important and there's ways that we can mitigate it. Blah 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 blah. But like huge tracts of the earth are like completely irreplaceable or like irreparably damaged. So like, might as well have some fun. Let's make some glowfish. I don't understand why people want to leave the planet. I don't get this. Like the let's go to Mars because the Earth is fucked crowd. Like Mars is awful. Like the ground is poison. Why would we want to leave a perfectly good planet that has a breathable atmosphere and rain and infinite water and all the things you could ever need? All we have to do is just stop breaking it, or reverse some of the damage that we've done, or build new ecosystems or whatever. It's much easier to do that here than it is to try and do that in space. Don't understand this. I've never understood the like manifest destiny and let's fuck off to Mars thing. It doesn't make any sense to me. Like it's cool, like from a science perspective, like it would make some interesting tech, but like why? Like if you ask an there's an astronaut who did, I can't remember his name, but like he did all kinds of interesting stuff where he ended up doing a mission into Antarctica, and like Mars is like Antarctica except without the water, and he's like, you know what, Antarctica is awful, but at the very least you can always go outside with a bucket and go get water, whereas on Mars, if you try that, you get a whole bunch of perchlorates and you fucking die. Like my one like nitpick with The Martian, even though it's a fantastic movie, is that the soil would have been poisoned and it would never have worked. That's the problem. Like the Martian stops making sense in the sense that the actual like soil, like the actual like dirt, dust stuff that he used to like make his soil to grow the potatoes wouldn't have worked. It would have poisoned the ever-loving hell out of the potatoes. They would have just died. Also, he would have died from breathing the dust. Okay, what else we got? Also, space stations sound fun up until you remember that space radiation is horrifying. So long exposure to that and you're going to be dead. Unless we're also down with genetically modifying humans to make them immune to that, but then everybody gets all fucking weird. So, yeah, just a simple, tiny, plot-destroying objection. It's a great movie, so I'm willing to, like, suspend disbelief for that one thing. But the fact that, like, it's two things. It's one, the soil is poison, and two, because he's, like, casually coming in and out of the hab, wearing that suit, that dust would get on everything and he'd die. So, most Mars spacesuit designs that are reasonable basically make it so that the suit never enters the hab. Like there's like a hatch of some sort that allows you to like leave the back of the suit and like decontaminate everything before you enter the actual hab, because otherwise you would die. Yeah, see, this is the problem. Analog, not Mars dirt. The analogs are useless because they always emit the perchlorates, of which there is large quantities of. Whereas the analogs on Earth match the rock composition, but they don't. They forget that the perchlorate needs to be removed. So they always do like a hand wavy, like, yes, you could grow it in there. Yeah, if you remove the perchlorates, which means you need huge amounts of water to clean your dirt before you can use it. Which Mars is kind of at a bit of a water premium, so doing this makes no sense. What do you think of ethics committees? I think they're a waste of time, mostly. I mean, like, this is why I do like private industry where ethics committees are basically non-existent. Like, because people are just gonna do whatever. Like, you can't actually stop anybody from doing anything. Like, if they want to do something ridiculous and stupid and awful, they're just gonna do it. Having an ethics committee doesn't actually stop any of that. Would you make a yeast with PETAs? Maybe not a yeast, but maybe a bacteria. I don't know why you would do it with yeast. What do you think of PETA? I mean, I like it with some hummus, but the organization is shite. They operate kill-only shelters. They kill 95% of the animals they take in. They're just an awful organization. Although even saying that could end up getting me sued because they're the worst. Anyway, yes, lunar dust will also kill you. Although, evidently not as bad because all the astronauts that we sent there were fine. But yeah, the dirt in space is generally death. Any book recommendations besides Noma and manga guides? I happen to like science books. Not really. I mean, I don't kind of no, I don't. Building Scientific Apparatus is another fantastic book. Yeah, I gotta readjust my chair here. Yeah, Building Scientific Apparatus is fantastic, and I'd highly recommend it. Yeah, space is death. Yes, absolutely. Although it was funny, I was looking at some. So the universe has been expanding, and as it's expanded, it's cooled. But if you rewind several billion years, there was a point when space was like room temperature. So, as long as you had oxygen, and I mean, you'd still need to, you'd need, a pressurized suit-ish. But, you wouldn't need to worry about, temperature regulation, because it'd be room temp. It's not, freezing cold like it is now. How does PETAs work? What's it actually break it down? What are the actual products? Oh, ethylene glycol. Yeah, that's not food. You would not want that. Yeah, so you end up with terphthalate and ethylene glycol. That sounds just wonderful. Do not want. Okay, can genetic engineering, modification, stop, reverse aging? Maybe. I don't know. Lots of people with lots of money are trying, but fuck knows. I hope so. I mean, I hope so in the sense that, I don't want to die. But I don't hope so in the sense that, if all the rich assholes who are already screwing everything up also don't die, that's kind of a problem. Because then you end up with, altered carbon. No. So there is actually a gene called damage suppressor protein that's taken out of tardigrades that basically makes cells essentially immune to radiation. The amount of radiation that a cell can take after this modification is just absurd. And they've done it. They've modified human cells with this thing, and then they irradiated the ever-loving hell out of them, and they were fine. They took, I think, 4 gray to the face, and they didn't even notice. Okay, what else we got? Couldn't you use the byproducts from plastic fermentation to create industrial products? Yeah, probably. Again, it doesn't need to be yeast though. You could just use bacteria for this. And if you added the right kind of bacteria, you could have ones that would take the ethylene glycol and terphthalate and just break those down as well. And use them as fuel. If you were probably to use R. palustris, this is the most versatile bacteria ever described. It can survive in essentially any conditions, or not quite, but it can do chemo this. It's Rhodopseudomonas pulsitaris or palustris. It's chemoautotrophic, photoautotrophic, chemoheterotrophic, photoheterotrophic. It can survive on oxygen. It can survive on carbon dioxide. It can survive on succinate. It can survive on all kinds of different things. It can break down. It can eat acrylamide because why not? Or acrylane. It can eat anything. It's really a very interesting organism. So yeah, if you were to combine this, and also there's been some efforts to try and genetically engineer them. So if you could make this do PETAs, then in theory you could just chuck bottles in and then turn it to carbon, basically. Yeah, legally bred. Yes, that is what that means. What jobs can one get with a bio or biochem degree? I am the wrong person to ask about this. Um, I, I've always maintained that there aren't that many. Like you can get jobs in academia, you can get jobs in various pharmaceutical companies, doing bio stuff. You can get jobs in the weed industry. You can get jobs in the agricultural industries. You can get jobs in various factories, depending on what they do. There's some synthetic biology jobs available as well, but they're harder to come by. Free online bio resources. I mean, there's a lot of good stuff on YouTube. Crash Course, Khan Academy, those sorts of things are good to get you started. Otherwise, it's just like books and learning the hard way, which is just like buy the materials, do the thing. It's the best way to learn. Most synthetic biologists I know are professors. Yeah, that's because that's one of the few jobs. Like 75% of bio grads end up not working in bio. And somebody sent me an email about this. They're like, I think you think that because they got they got degrees in ecology. I'm like, no, like molecular biology. Like unless you get a job in academia or in private industry, like there's not that many bio jobs. Although it's been very funny that all of a sudden everybody really appreciates the biologists. When before they were like, fuck you, make coffee. Now they're like, oh God, please save me. I'm like, yeah, well, should have provided funding for research then. Could we use the resilient against radiation from 𝐎𝐵𝐺 𝐸𝐴 for something interesting, and how does it work? I have no idea how their radiation resistance works. The only radiation resistance I'm familiar with is the thing from tardigrades, which is damage suppressor protein, and I happen to have that one in my fridge. Just for those who've been wondering, and I intend on modifying some human cells and making them hella radiation resistant. I'm going to just expose them to a shitload of UV and see what kills them. What was the specific issue with the styrofoam eating worms? The bacteria that was supposed to be inside of them never grew. What superpower would you want? I mean, the like, could I go with the Deadpool regeneration, but without cancer? That sounds good. I'd take that. Make a gene gun as a video. Yeah, that's the plan. Alright, let's do one or two more questions, and then I'm going to wrap it up for the day. Just as a reminder, the next stream will be on Monday, same time, 2 p.m. Eastern Standard Time. And if you've got ideas that you'd like to submit to the show, to be added to the hat, send them to me on Twitter. Use the hashtag #Who'sGene. For the synthetic opal project, is it possible to make hydrophone opal? I have no idea. Honestly, I have no idea. Maybe? I don't know. Can we make a unicorn? I have no idea. That'd be messing with like Hawk's genes and stuff, and it'd be hard. If given a high tier lab and immense resources, what project would you like to make? I don't know. I could probably make some like superhero serum. Probably. I don't know. Like bioluminescent plants. Working on it. Can we make blue roses? I've got a friend who's working on it, and I'm gonna have him on as a guest. Oh, that's the other thing. So I'm working on getting some of my really cool bio friends on as guests for future episodes. So I'm talking with some of the people from Open Insulin, so that'll be fun. I've got my friend who does some really cool plant work and who's doing like rainbow flowers and like blue flowers and that kind of thing. And so he'll be on at some point. Yeah. So that'll all be fun. No, not Hawk gene, Hawks as in HOX. Although technically horns are a weird form of bone cancer that things with horns figured out how to like keep in check, which is just so weird and wonderful. I love bio sometimes. I mean, this is that kind of stuff is the reason I love bio. When I'm sitting in a lab bench is one of the reasons I hate bio. Do you plan on doing any industrial production of products? No, that's not my thing. Make a dragon. Yeah, all right. Are there any benefits in changing the rose's color? No, it's just cool. Any YouTube recommendations? Okay, yeah, actually, this is fun. Let's do this. And so I'm gonna do this, and then I'm gonna wrap it up for the day because I'm hungry. Okay, so YouTube recommendations. Some channels that I really like. 𝐎𝐫𝐢𝐠𝐢𝐳, that one's fantastic. Applied Science, of course. Nighthawk and Light, of course. Who else? And it's I feel a little weird doing this because I feel like I'm gonna end up forgetting somebody, and then I'm gonna feel stupid for snubbing them. But that's not my intention. I'm just kind of going off my head because I didn't like prepare a list or anything. What else? What other channels do I really like? ClickSpring is fantastic. What other science channels do I watch? I don't know. Let me have a little look see and see what channels, because I know I've got a really fun one is called Crime Pays But Botany Doesn't. I would highly suggest that one. It's very funny. It's this dude from he's got like a thick, I want to say Chicago accent, like blue collar Chicago accent, and he knows just an enormous amount about plants, and he just like fucks off to a forest and he's like talking about plants and stuff. It's awesome. Nautilus Live is a really good one as well. It's basically a bunch of bio nerds on a multi-million-dollar vessel with like an underwater ROV, and they're like exploring the bottom of the ocean. It's magical. What else BPS Space does really cool rocketry stuff. I'd highly suggest giving them a watch. Extractions and Fire, or Explosions and Fire rather, is also another fun one. Journey to the Microcosms is fantastic. PBS Spacetime is great. What else? Tech Ingredients is good. Does a lot of really cool stuff. If you want like more of like a maker channel that just like makes interesting weird shit, 𝐎𝐫𝐢𝐠𝐢𝐳 is very fun. He does a lot of stuff with just like hand tools, but makes really cool projects. If you want like a food channel that's just like aggressively relaxing, which is sort of I realize how that sounds, but like you'll see what I mean if you watch it. 𝐷𝐸𝐠𝐸, it's this Chinese woman who is like off in the mountains in China, and she's got like her whole little farm thing, and she just makes aggressively pretty videos, and like food that looks just absolutely incredible. So highly suggest that one. What else we got? I'm trying to see if I've got any other like hidden gems that I just haven't seen in a while. If you want just like funny animations, Harry Partridge is really good. Of course, Jerry Ellsworth, although she doesn't really make a whole lot of stuff now because she's busy with her company, but her channel was always great. What else do we got? 𝐎𝐫𝐢𝐠𝐢𝐳𝐚𝐥, of course, very fun. There's another channel which he's a friend of mine, just Chris Bowden. He does a bunch of really cool engineering stuff. He used to run the Geek Group, and so that was really cool. But that got shut down. Okay. Yeah, Smarter Every Day, Veritasium, of course. Yeah, those ones. Those are all pretty great. So yeah, I mean that'll hopefully give you something to do while you're bored in confinement. Can you make a detailed plasmid design tutorial using Benchlink? That's literally what this stream is. Just it's the whole point of this stream is to see how this works, because this is the whole tutorial. Just do the things I'm doing. I mean, yeah, Nile Red, Nerd Rage, of course. Their channels are so large. I just assume that everybody knows them. I'm trying to think of ones. I mean, 3 Blue 1 Brown. Like I'm trying to think of the smaller channels. Although, admittedly, some of the ones I listed off have millions of subscribers. But I mean, they're all fantastic. Oh yeah, Tested and Adam Savage. They're, of course, fantastic. Vsauce. All that kind of stuff. Do you have a pro Benchlink account? No, I've got the free account. Favorite anime? Okay, this is going to be my last question because then I really need to go have lunch. My favorite anime currently, Shokugeki no Soma, or Food Wars, is fantastic. So that's a cooking anime. Think Iron Chef meets Anime High School. It's fantastic. Literally some of the best food I've ever eaten. Another good one is called Gate. Oh, somebody pitched in. Oh, thank you, Mikey. Do you have any advice on PPE? Yes, wear as much of it as you can. No. N95s, don't touch your face. Don't touch the mask. I swear to God, if I see one more person wearing a mask over just their mouth, I'm going to fucking lose my mind. What else we got? Oh yeah, so anime. Gate is a really great anime. It's just go watch it. It's great. Dr. Stone, I've really enjoyed. One Punch Man. I don't know. There's a lot of I don't really watch a lot of anime anymore. Yeah, there's certain channels that are being suggested which I'm explicitly not. Yeah. So those are. Oh, Moyashimon. Yes. Oh my God. Yes. I love that. I love that anime. I'd totally forgotten about that one, but that one was really cute, and I liked it, and I learned a lot about fermenting. It's such a weird anime. I love it. It's so good. Anime is bad for your emotional development. Clearly spoken by somebody who hasn't watched good anime. Yeah, Tom Scott's a great channel as well. else do we got? Okay, well, I think I'm gonna wrap it up for the day then. This has been a lot of fun. Again, we're gonna do the next episode on Monday. Ideally, if all goes well, and I manage to get it all done, I should have an actual like video out in the next couple of days. I'm still undecided on whether or not I want to do one for Thought Emporium or one for Taste Emporium first. I'm working on one for each. I'm thinking of Taste Emporium first just because it's basically filmed and ready to go. So I'm thinking that'll probably be out in the next couple of days, and then there'll be a Thought Emporium video out shortly thereafter. But then yeah, that'll be coming out. I hope to see you guys at the next stream on Monday. Again, if you've got ideas for things that you want to see us design, be sure to leave them on Twitter. They'll be added to the hat. Check the, again, as a reminder, please go check out the plasmids that are on GitHub and put in any corrections if you think that there are corrections needed. But other than that, I will. That's where I think I'm going to leave it for now. I hope you've enjoyed. I'll see you next time. Have a good day. Stay safe. Stay healthy. Don't touch your face. See you next time.